Great, thank you. And thanks Renzo, and thanks GSC. So the reason we all come to meetings like this, the reason that I hope all of us, but certainly most of us are in this room, is because we believe that, that science will progress most effectively, most efficiently, and most rapidly if we live in a world where data, scientific data, flow freely, move around the world easily, are made as available as possible, and most importantly, are described as richly as possible. And so there are two big components to this. We've heard a lot about the minimal reporting requirements, the MiXs checklist. Another component is the existence of long-term data repositories into which one submits information, the scientific data points themselves that are described by the minimal reporting requirements. So what I'd like to do is talk, now I only have a short time, I can't possibly talk about all the different submission routes, so I will allude to those routes in the how part of this. A little bit of, there are some complex incentive schemes, incentive landscapes for individuals in the process of submitting and describing their data. I'll touch on that. And then I'll come to talk about the direction of some of this and introduce some areas that may not be so obvious that I think will make a difference to the way we think about submissions, the way we think about the flow of information in this area. So just to start with, some context, the International Nucleotide Sequence Database Collaboration, the INSDC, is a well-established, so it's been operating for the best part of 3 decades now, a well-established collaboration, data exchange collaboration that operates at the European Bioinformatics Institute, that's my affiliation, the National Center for Biotechnology Information in the US, in Washington, the NCBI, and the DNA Data Bank of Japan, so that's in Mishima in Japan. And so this 3-way collaboration collects the world's public domain sequence information and organizational information around the sequence and interpretation of the sequence, and makes it available for everybody to use in the scientific community. The information that we capture is diverse. There's a spectrum of information, so we go from fairly rare, comparatively raw forms of information, so sequence reads, almost as they roll off sequencing machines. We have layers of analysis and interpretation of those raw forms of data into higher order structures, so alignments and assemblies and OTU clusters and so forth. And then we have submitted functional annotations. So this is where the scientist is asserting a particular biological function for the region of a molecule that's been sequenced. And then what we do in this system is we provide ways of capturing the world's sequencing output into the system. So we provide local submission tools at each of the partners' sites in the collaboration. And we work with third parties to provide submission tools as well. And then a lot of the work is this. It's about exchanging data between ourselves. So the very heavyweight moves of data around the world on a daily basis. When we have all of this information, we try to organize it and we try to make it available in the most useful ways. So we present browsers and tools to search and to discover. Increasingly, we focus on providing programmatic services as well and various tools to do analysis. So you can, the ideal is that you can get all of you can find the data sets you want and you can get to those data sets easily. And of course, there are peripheral activities that we need to get involved in, helping users to use the system, training users, developing technologies, and working with groups like the GSC. So members of INSDC partners have been involved with Genomic Standards Consortium right from the beginning and will continue so to be. So Palin has already introduced the MiXs standards, a family of standards, and explained how by slicing in by type of organism, by taxonomic area, by application, and by environment, how you can pull out a checklist for different studies. And so what I'm going to talk about is how we are implementing, or how providing this richer information impacts the different submission systems that we offer. So submission, as we've heard several times, it doesn't happen without any effort. And the purpose of what we do in designing submission tools is to lighten the load and to make things easy, but inevitably there is some effort required on the part of the person who has the information to impart that information into the repository, or to describe it to another scientist who's interested. And so I have a mantra, and I very much like other people to share this, and it's that when you're asking a question of a submitter, of the provider of data, you have to ask that question at the right time. You have to you certainly have to be asking the right person. You have to ask the right question, otherwise you may not get the right answer. and you have to ask the question in the right way. And so the right time is important because information, unless it's stored in a very systematic and organized way, it tends to decay and becomes less available. So my lab notebook is more useful to me now than it will be in 2 years' time because the organization system is in part not written down, it's in my head still. And I think that's true no matter how good we are at recording things. The right person, the best person to ask is the person who's actually done the work. So if I've been out on a ship sampling something, then I'm the person that knows what I've done. I can tell that to somebody else, but that information, the message may be a little bit degraded. So we need to get to the right person. We need to ask the right question, and that's where the checklists are so important, because although as a provider of information, I have all these interesting things I can tell you about it, that doesn't necessarily marry up with what ultimately the consumer of that information wants. They will have a certain set of questions that I may not have foreseen or predicted. And then you have to use the right method. You have to ask the right question in the right way. So here, if this is in a computer, then I need to have some kind of programmatic interface. If this is a huge load of data, I need one type of interactive interface, perhaps. If it's a small thing, the person providing the information is only ever going to talk to me for 5 minutes, then perhaps I ask them directly, or I provide a much more intuitive interface that is much easier to learn how to use. So the submission tools abound. There are many submission tools, and it's simply because we're trying to provide for all these different people will fit into this system. Different providers fit into this scheme in different ways. And so if I'm a sequencing center, I need a different interface for submission to somebody who's on that boat sampling. And so I won't list all the tools. We've heard a lot about SRA Webbin. We've heard a lot about ISA. Those are the tools that are closest to EBI. There's a much bigger list. This includes the tools that are closest to EBI, NCBI, DDBJ in the middle, and then NCBI. What I would recommend is that the starting points are these URLs where you can get into each of the contributing systems, and you will then be triaged appropriately. And just to look at some of those incentive schemes, those incentive landscapes for people doing this, and I think it's important to understand a little bit where people come from. So overall, we know that I'd like to be able to read this here, but if I can, maybe I can just, it's got bigger. So we know that there are many benefits to sharing data. This is why we're all here. You can discover the data more easily. When you've discovered the data set, it's more useful. You can do more things with it, do more analysis. You can reproduce results. You can reanalyze. You can meta-analyze. And increasingly, you can bring things together data sets together that weren't initially combined, and you can look at emergent properties, and you can bolster your statistical power when you're doing an analysis. So these are great. If we look locally, then the benefits to the person who actually has to provide the information, then although morally there is a duty to provide all, there is a benefit, an overall benefit to providing all the information and complying, perhaps it's the more local drivers that have an impact. So increasingly institutions require submission of data. The spirit of the policy around these requirements includes the requirement that data be well described. So I think the GSC has work to do to continue to push for these various policies to include specifically and explicitly lists of checklists that should be complied with. And the same funders have similar policies, and that's a growing area, and publishers as well. It's completely accepted that people submit data to repositories when they're publishing a paper. The spirit of that is that the data need to be sufficiently described. So this is an area for GSC to work in an ongoing way. Of course, immediately one can share data with collaborators in a more useful way. Data are expensive to store. If we describe them properly, transfer them early to public repositories with the appropriate compliance information, then this is more useful to partners, even ahead of publication. But of course, if I publish my science early in a rich way, in a well-described way, it's more useful to people, and I promote my own area of science. And of course, ultimately, this means there will be more citations. So I think I have to skip the first of my changes that I was going to mention. Suffice it to say that technology is always changing, the network availability and connectivity, the portability of equipment is allowing us to build different modules of submission tools and to ask those questions in the right place. We're much better able to ask the questions in the right place than we used to be because people have machines with them and they can capture information. And also, because things are more connected, we can immediately connect to satellite information and we connect to other databases for information. A change that's taking place across those of us or across institutes who provide large-scale data repositories, so particularly EBI, NCBI and DDBJ, is that while for many years individual resources grew up, so data resources such as sequence databases, expression databases, genome databases, proteomics, peptide identification databases, grew up as individual freestanding projects where you had a database and a submission tool or a set of submission tools, and the provider would interact with each of these tools. Increasingly, we're moving to a world to make us more efficient and to make us provide better services, where we actually provide more specialist data-focused data type-focused databases. And then we provide more modular submission tools, where for many of the assays we're doing, we need information about the experiment that's been done. We need information about the sample. We might have some functional annotation, and then different types of data. And so increasingly, the user, the provider, will face a different combination of these modules when they're working with the submission environment. And these will be presented at different times in different places. So finally, this is something that I spend a lot of time concerning myself. It's about the very rapid and relentless growth of sequence data. What's absolutely clear is that unless we have and deploy some technology to compress data in quite, I hope, sophisticated ways, we will not be able to store all data sets. And so I've been involved in a technology called CRAM, which is a compression technology that allows you, it gives you a volume control. You can compress sequence data more or less. Now, when it's more, it's lossy compression. You have greater compression, so you lose, hopefully not too much signal, but you do lose signal. When the volume is down, you lose nothing. So this is a compression that allows you to be selectively more or less aggressive about the amount of compression you apply to different data sets. And so the community, the scientific community as a whole, needs to turn to thinking and making decisions about where we focus the most aggressive compression, which data sets warrant very heavy compression and greater loss, and which data sets really need to be preserved. And so think about this in economic terms, and there's a paper that we've just published recently in Gigascience. Think about this. We have two axes. We need to consider the cost to replace the sequence. Now, if we do something to the data set that makes it less useful, we need to replace it somehow, if we ever want to get back to it. That could mean resequencing a stored sample. It could mean rerunning an experiment on some physical sample, but it could mean going all the way back and resampling. And so there's a huge cost involved in, depending on the nature of the sequence and the thing that's led up to the sequence, a huge cost there. But we also need to consider the value of or the usability of the data. Once we have a data set, if it's not richly described, if it doesn't comply with the community standard to describe it, then it is less useful. So overall, it has less value. And so we can think about partitioning data sets into different bins and afford them different levels of compression. And so for those data sets that are up here, we're going for lossless, so this is the volume control is set to low. So this is a high cost to replace, but it's well described. So it's very useful. And this is a lower cost to replace, but it's very useful, so we put it up here. So we're going to apply 20-fold compression. Here we have a high value sample. It will be difficult to replace, but it's not well described. It doesn't comply with any standards, so it's not actually very useful. So we probably have to go and repeat a lot of work anyway. And then finally, the heaviest compression for the low value, the low replacement cost, where there's a poor description. And so really, rather than think about this as well, the better described your data are, then the more we will preserve them. And this is I stress, a community decision. It's not a repository decision solely. Rather than say, the better you describe, the less we compress. I think we really have to say that if you don't describe, then there really is no value in storing. So we have to be quite binary about this. So I'll just acknowledge the many people, or the institutes of the many people involved in this, and I'd be happy to take any questions if there's any time.